Okay, great. Thank you so much. And thank you to the organizers. I first came to India 20 years ago, where I first met Kumar. And it's really been a pleasure. I've been here multiple times since then, but it's really been a pleasure to see how India has changed, the progress that's been made, and have something as beautiful as this convention center is really quite interesting. Okay, I'm going to talk today a little bit about some insights that we're gaining into disease mechanisms based on genomic studies for mainly primary open-angle glaucoma. These are disclosures, none of which are relevant for today. So I think you all know, and I'm not going to spend a lot of time here, but glaucoma is an important cause of blindness throughout the world. And importantly, early detection and treatment are really necessary to preserve sight in affected people. But disease screening and risk stratification are challenging. It's hard to identify people who are truly at risk for disease before they develop damage to their optic nerve. And as a result, many affected individuals are undiagnosed. So that's really the premise for why many of us in this room have been engaged with studying genetics and glaucoma for many years now. Glaucoma turns out to be one of the most heritable of all human diseases, and we've had a lot of success in GWAS and other kinds of genetic analyses. There are now more than 300 genes contributing to disease risk that we've identified from these kinds of studies. And so I present a little diagram here as we're beginning to understand pathways and processes that these genes fall into. We are beginning to identify some of the mechanisms that could be underlying disease in glaucoma that's really being illuminated by these genetic studies. So today I'm going to give you 3 examples. I can't obviously talk about all 300 of these genes or even all of these different processes that have been identified through our understanding of what these genes do, but I want to give you 3 examples just to shed some light on kind of where we are in terms of our investigations and what the future could look like in terms of understanding the landscape. So the first one I want to talk about is mitochondria. You all know that mitochondria is the powerhouse of the cell. It's the source of ATP for cells. It keeps cells alive. Mitochondria is particularly important in glaucoma because the retinal ganglion cells that are really the target of the disease process have a high energy requirement and are very dependent on mitochondrial function. So one of the things that's emerging is that the mitochondrial electron transport chain, which is necessary for ATP production, the first complex of that chain, complex 1, has 45 different subunits, so it's a very complex subunit. But 2 of these subunits have now been implicated in glaucoma. I'm going to give you a little bit of data on each of those. So the first is NDUFS3, which is an important protein that forms part of the complex. This is a nuclear encoded gene that then is transported to the mitochondria to function. And this gene has been associated now with POAG in gene-based analyses from the last GWAS that's been published, Han et al. In Nature Genetics, with a very significant p-value. So this indicates that dysfunction of complex 1 leads to mitochondrial dysfunction and potentially then increases susceptibility to glaucoma. The second protein subunit of complex 1 that's interesting is also a gene that's nuclear encoded, FASKD1. And here, this data comes from an exome-wide study that we carried out, and we found 2 independent rare missense alleles in this gene, both showing nominal association, but significant effect sizes. And this is a protein that regulates a subunit of complex 1. Decreased FASTK1 leads to increased mitochondrial ND3, which is one of the subunits, and increased activity. And this has come from some functional studies carried out in HEK cells using CRISPR. And I also just showed you here the single cell sequence data for both the MDUF-S3 as well as the FASTKD1 in retinal ganglion cells to compare their expression in these different types of ganglion cells, and also to TXNRD2, which is another mitochondrial protein that's involved in glaucoma. Now, the second example that I want to show is CAV2. So one of the first loci for primary open-angle glaucoma was an association with SNPs that are located in a non-coding region between CAV1 and CAV2, which are sort of arrayed in a head-to-tail fashion on chromosome 7. There's always been a lot of attention to CAV1, but recently, CAV2 has also been associated with primary open-angle glaucoma. And we have identified, also in our exome study, a missense allele in CAV2 that is highly associated with disease. What's interesting is that these CAVs, both CAV1 and CAV2, and also CAV3, are involved in formation of these very specific molecular outpouchings in membranes. And there's a number of different biological processes that occur at these membrane pouchings, including regulation of NOS3 and intracellular cholesterol trafficking. So involvement of these two caveolins argues that this is an important mechanism in glaucoma and one that warrants further study. And then the last one I want to show you today is also getting at a little bit of something that Dr. Heise talked about as well, and that is this TEC pathway with the ligands angiopoietin-1 and angiopoietin-2. We know that mutations in TEC and mutations in angiopoietin-1 causes severe early onset glaucoma, like congenital glaucoma, in some cases, in some families with variable expressivity. And this is due to the fact that losing the function of this pathway leads to abnormal development of Schlemm's canal. So Schlemm's canal doesn't form, fluid can't get out of the eye, and the pressure goes up. So what we found a few years ago is that there's also an interesting SNP in angiopoietin-2. So angiopoietin-2 had not yet been implicated in glaucoma, and we found that this SNP in angiopoietin-2 is protective. And we also found, using data from the UK Biobank, that the protective SNP reduced intraocular pressure. Now, what's interesting about this SNP is that it lands in a microRNA binding site. And so this change from the G to T at this SNP could actually disrupt binding of the microRNA to the 3' untranslated region of angiopoietin-2, which would then increase expression of angiopoietin-2 because binding of the microRNAs can depress expression. And in fact, what we found is that the protective allele is associated with increased expression of angiopoietin-2 and decreased intraocular pressure. But the really interesting study was one that was just published this past fall in collaboration with Susan Quagen, our collaborator. She made a mouse that actually has this same mutation in it and showed, in fact, that the level of angiopoietin-2 is higher in the mouse tissue and that the Schlemm's canal is larger and better formed in the mouse that has this variant. So this points to this kind of important TEC signaling strength where we have TEC mutations that cause early onset disease as well as adult onset disease, which is interesting in terms of thinking about the full spectrum of glaucoma disease in some of these genes. So just to summarize, genetic studies are identifying hundreds of genes related to pathogenesis and disease risk. Protein function suggests novel disease mechanisms. I can't emphasize enough that the functional studies are very important to show that, in fact, these mutations are really having an impact in defining the mechanisms, and that these mechanisms are important if we're going to think about how gene-based therapies could be used for these diseases. I'm just listing here all the people who are in the International Glaucoma Genetics Consortium because that's a huge source of the data for these experiments, as well as people who collaborate with us on these different projects. Thank you.
And we're open to questions.
In addition to the complex one, 45 subunits told you, some of the new genes, especially that is involved in the mitochondria fusion and fission. So some of the genes are very, very important. Did you have come across any genes associated with the mitochondrial dynamics?
Yes, actually, there is a gene involved in fusion and fission that is also associated with glaucoma. Yeah. In a study that I did, where it was very simple, I just took all the genes that are in mitocardia, which is for the mitochondria, and then all the genes that were significant in gene-based testing from this most recent GWAS and just looked at their overlap. And we found 16 genes that overlap between glaucoma and the mitocardia. So in another talk, I would talk about all of those.
Thank you.
But the end of the end of S3 is a really interesting gene because we know that it does have it's already a cause of Leigh syndrome if you have 2 copies. So here we're talking about a change in expression that then predisposes to glaucoma.
Great talk, Janey. My question is somewhat related, but were there corresponding mutations in the complex one in the same patient who also had mutation in the nuclear gene?
That also have what?
Corresponding mutation in the nuclear gene that actually hosts for mitochondria.
Oh, well, those studies came from different data sets, so you wouldn't be able to look at the same variation.
So I have a question, Janey. So
Yeah.
it's very interesting work. So you showed 2 genes of mitochondrial nature, and you said that is with the ganglionic cell layer, right?
The what?
In the ganglionic cell layer?
Yes, right, yep.
So does it have any relationship with the IOP,
With myopia?
with intraocular pressure?
Oh, yeah. So an interesting thing is that in the GWAS, the one before the one that's in preparation, there were 5 different genes in that GWAS that were associated with intraocular pressure that were mitochondria genes. So, and we know that one gene that's a mitochondria gene that's now been in multiple GWASs, TXNRD2, that's involved in NAD metabolism. And we know that gene is expressed in the anterior segment and in the posterior segment. So, it might be one gene from the mitochondria side that impacts both ganglion cell health and intraocular pressure. Yeah, certainly there's a role for mitochondria in the anterior segment, yeah.
Thanks, Janey.